From the tiniest insect to the largest elephant, every creature shares one incredible thing in common: DNA. DNA is present in all beings and is the genetic material passed on from parent to offspring for all life on Earth. DNA holds the complete set of instructions needed to build and maintain an organism. The building blocks of DNA are nucleotides. Each nucleotide consists of a phosphate, a sugar, and a nitrogenous base. There are 4 types of nitrogenous bases in DNA: cytosine, thymine, adenine, and guanine. These bases are complementary with each other. Adenine pairs with thymine and cytosine pairs with guanine. It is essential for the DNA to replicate because when a cell divides, each new cell has to have a complete set of genetic information. This is done with a process called DNA replication. DNA replication starts at the origin. An enzyme called the helicase unwinds the DNA. Then, another enzyme comes into play and is called the primase. The primase makes these RNA primers on both strands, which serve as starting points for the DNA polymerase. The DNA polymerase is the most crucial enzyme, and it starts to build a new DNA strand starting at the RNA primer through the 5' to 3' direction. Once DNA replication is complete, cells also need to use DNA to make proteins. And this is what's called protein synthesis, and it mainly consists of 2 parts: transcription and translation. And it begins with the process called transcription. Transcription is the process of making a messenger RNA, aka mRNA, copy of a gene. It begins when the enzyme RNA polymerase binds to the start of a gene. Then, the RNA polymerase unwinds a small section of the DNA and uses one strand to build the mRNA. Instead of thymine, RNA uses uracil, so during transcription, adenine pairs with uracil during this process. After transcription, the mRNA leaves the nucleus and goes to a ribosome, where translation takes place. Translation is the process of turning the genetic code in mRNA into a chain of amino acids which will fold into a protein. The ribosome reads the mRNA 3 bases at a time called codons. Each codon matches with a complementary anticodon carried by a transfer RNA. Every tRNA also carries a specific amino acid. For example, this one is carrying histidine. When the tRNA anticodon pairs with a codon on the mRNA, the ribosome helps link the amino acid it carries to the growing chain. This continues codon by codon as the ribosome moves along the mRNA, with new amino acids being added one at a time. Once a stop codon is reached, the chain is released. The chain of amino acids then folds into a protein which carries out the most important functions of the cell. And that's the amazing journey from DNA to proteins, the instructions of life in action. Thank you so much for watching, and I hope you found this video helpful.